We're talking first about the induced fit of an enzyme. We used to think that enzymes worked more like locks and keys. So here's the idea. Here's a doorknob. Whoops. And here's a key. It's the opening. Here's the key. And the key had a very specific shape, and it was only this shape that could unlock that particular keyhole. Pretend it's the right size. Um, and that's really probably not how enzymes work. This is the induced fit model. And if you look at the active site here, this does not look a whole lot like this. So the substrate actually has to come into contact with the enzyme in order for the enzyme's active site to change shape so that it does fit just perfectly. So let's take a look at that. A change, and you can see it right here. So you start out with the enzyme and the substrate. The substrate gets in here and it changes the shape of the enzyme's active site. And there's the catalysis where the substrate breaks down to make the products. And those are the products. Don't get confused here with the term substrate. The term reactant could also be used. So in a chemical reaction, we have reactants. Either one reactant in this case, or sometimes reactants. And they either break down or get put together to make the product or the products. And then we're just going to put products. In this case, because there's an enzyme, the reactants can also be called the substrate. So we're going to use these two words interchangeably when an enzyme is working. Substrate. And so here you have the reactant, which is also called the substrate. In this case, there's just one reactant or one substrate. And then let's see it break down. And there it is. Now you have the products that are being released. So a change in the shape of an enzyme's active site is induced by the substrate. So that's the induced fit model as opposed to the old-fashioned lock and key model. What affects enzyme activity? I'd like you to put a few stars next to this because this is so important. And it's the first factor here that we're going to look at mostly. So there are three factors. The first one is environmental conditions. And then there are cofactors and coenzymes. And there are enzyme inhibitors. So the environmental conditions is what's really most important for you to remember. So let's look at those first, and then we'll spend just a couple seconds on cofactors and enzyme inhibitors. Environmental conditions. There are a few environmental conditions, or a couple environmental conditions that you should really memorize. The first one is temperature. So the idea is that enzymes work best at a certain temperature. If it gets too hot or too cold, the enzymes just stop working. So high temperature may denature or unfold the enzyme, and so hydrogen bonds come apart. So here's a new vocabulary word. Maybe you can put a star next to this too. You need to memorize that. Denature means to unfold. So remember that an enzyme is a protein. And remember that proteins have this special shape. It's amino acid to amino acid to amino acid. And you can have a long, long, long chain of amino acids. The order of the amino acids is super important. A different order means you'll have a different protein, which in this case would be a different enzyme. So that's the primary shape of an enzyme. Now, if I don't draw all my little circles, I can show you the secondary shape pretty easily. So here would be the secondary shape, and you're imagining that there are a bunch of amino acids strung along here, and that this chain is made out of amino acids. This chain is made out of amino acids, and I just drew it a little bit more magnified. So you can get these alpha helices or these beta pleated sheets. That's the secondary shape. And then if this folds over on top of that, you might get something that looks like this. That would be the tertiary shape. And then if you have this with another one, that would be the quaternary shape. So anyway, the shape of the enzyme is really important because let's say that the active site is right here. That actually came from a little bit here and a little bit here. So if you take this thing and you unfold it, whoops, I don't know what that's doing there, but anyway, if you take this thing and you unfold it, you're going to get something that looks like this. And so the problem is that right here and right here are not near each other. So how do I get rid of this thing? All right, let's go backwards. Oh, now my picture's gone. Wait a minute. There's my picture. Okay, so here's what the active site looks like. And if you unfold it back to this secondary structure, the active site here is spread apart to here and here, so the substrate can't get in there and change and get broken down or put together. So a high temperature will denature or unfold the enzyme, and then it won't work anymore. You can have a temporary denature that can fold back up the right way, and sometimes you can have, so that would be reversible. If you have an irreversible denaturing, then it just gets unfolded and it cannot go back together again the right way. So here's a graph. You should really, really, really plan on trying to get used to these graphs. You need to be able to read something like this for me. So here's the rate of reaction. That means there's pretty much no reaction going on here, and there's a lot of reaction going on here. Here's the temperature, and so it says that down here. This is 30 degrees Celsius. Here's the Celsius part. Here's 40 degrees, here's 50 degrees. Body temperature is around 37, I think. So if you look at this, about body temperature here is about where this enzyme works best. If it gets too hot, the enzyme denatures, and if it gets too cold, the enzyme denatures. So in the human body, it wouldn't get that cold or that hot, so the enzyme would always be working. Another one besides temperature is pH. Let me just go backwards for a second and talk about before I go on. If it gets too hot, it's probably because the enzyme's denatured. If it gets too cold, it might be because there's not enough activation energy, or maybe it's changed shape a little bit as well. Okay, so the next important one is pH. Most enzymes work best at pH 6 to 8, which is near neutral. So remember the pH scale. It's a super important scale. It goes from 0 up to 14. The middle is 7. And if you just remember your alphabet, A, B, C, D. So A is for acids. Acids are 0 to almost 7. And B is for base. That's 7 point whatever, anything, up to 14. So pH 6 to 8 is near neutral because pH 7 is neutral. And so the reason that most enzymes work well at this neutral, close to neutral pH is because your body is mainly at a neutral pH. Here's a really interesting graph of two different enzymes that are in your body. One is pepsin, and the other is trypsin. These are both enzymes that break down proteins. And again, we're looking at rate of reaction, so this means that down here there's not a very much of a rate of reaction. In fact, the reaction's not going at all. Up here, you would get a very, very fast reaction. So if you look at pepsin, its highest rate of reaction is 2 point, I don't know, 2.5, 2.6, something like that. That's a really, really low pH. Look, here's neutral at pH 7. So where in your body do you have a really, really, remember A comes first, where do you have a really, really acidic environment? Oops. I hate when so I think I know how I'm doing that at least now. So where in your body do you have a really, really acidic environment? In your stomach. So, you know, when you throw up, it burns your throat and it's really gross. That's your stomach fluid, and it's acidic. So pepsin works in your stomach, trypsin works in your small intestine, and look, it works very, very close to pH 7, very, very close to neutral. There's just a little bit of overlap here, but the optimal pH for trypsin is close to neutral, 6 point something, and the optimum pH for pepsin is very, very, very acidic, 2 point something. So those are the environmental conditions that you need to know. Cofactors and coenzymes. These are inorganic substances, so the cofactors are inorganic substances. Inorganic means there's only one carbon or no carbon at all. So these are minerals, stuff like zinc or iron. And so you might need some iron, maybe an atom of iron in this particular enzyme in order for it to work. Or vitamins would be the coenzymes. And for me, I don't really care if you know the difference between them. But the idea is here you have something that the enzyme needs in order for it to work the right way. So cofactors and coenzymes are inorganic substances like zinc and iron, or vitamins that are organic, that sometimes that are sometimes needed for proper enzymatic activity. So in other words, if you're really, really deficient in zinc, you'll have some enzymes that don't work the right way. If you're really, really deficient in, let's say, vitamin D, again, you're going to have some enzymes that just don't work very well. And then the last thing that affects enzyme activity that we're going to be talking about are enzyme inhibitors. So here's a vocabulary word that you need to know. To inhibit something is to subdue it, to decrease it, to make it stop or at least slow down. Another term that we use a lot is prohibit, which means to totally stop. But inhibit just means to slow down, maybe to very close to stopping. And so let's look at those. Competitive inhibitors, when you think of competitive, so let's say you're running a race and it's a competitive race, and either you're going to win or the other guy's going to win. So a competitive inhibitor is a chemical that resembles an enzyme's normal substrate and competes with it for the active site. So let's take a look at that. Here's the enzyme, and let's go backwards a second. Remember, I showed this picture before, and this is the active site. So this is where the substrate or the reactant should go in order for it to get broken down to become a product. But look, this thing is a competitive inhibitor. It sits right where the substrate's supposed to sit. So now that substrate comes along, and if this thing were not here, then the substrate could go in and get broken down and the enzyme would work. But this thing is here, and so now the substrate can't be broken down or put together. Oops, there's the substrate. So oops, can't go in. Non-competitive inhibitors. So these are ones that are not competing for the active site. So these are inhibitors that do not enter the active site but bind to another part of the enzyme, causing the enzyme to change shape, which in turn alters the active site. So let's take a look at that. Here you have that enzyme again, and remember this thing is supposed to look like this for this particular pretend enzyme. But it doesn't look like that because here's this non-competitive inhibitor. Whenever this thing is stuck here, this enzyme's active site changes shape. So now the substrate comes along and oh my gosh, it doesn't fit in there anymore. So now the enzyme doesn't work. So the enzyme, if the enzyme doesn't work because there's something stuck in its active site, that's called a competitive inhibitor. If the enzyme doesn't work because there's something stuck somewhere else that messes up the active site, then that's called a non-competitive inhibitor. So your body uses inhibitors sometimes to regulate an enzyme, to say, okay, I don't need you right now, or let's get rid of the inhibitor, okay, now I do need you. Or sometimes inhibitors are something that the body doesn't produce and doesn't want there, but it's there because there's something toxic in the environment, for example, or there's a virus or something there. Okay, quiz time. Oops, 13 minutes. I think I'm going to pause right here. Let's just do this first one. Enzymes are usually what type of molecule? Enzymes are proteins. Enzymes work by lowering activation energy. List some things that can affect enzyme function. So hopefully you can remember pH and temperature. Those are the important ones for you to know. Those are the environmental conditions. If you also remember having coenzymes or cofactors around, that's great. And if you remember the lack of inhibitors, then that's another one that would be good.